4 billion trees didn't die. That's the number people get wrong every time this story comes up. 4 billion American chestnuts vanished from Appalachian forests in under 40 years. And yet, right now, under the leaf litter of 100,000 acres, this tree is still alive. Not extinct, not resurrected. Something stranger, suspended. For over a century, the American chestnut has been running the same doomed loop underground. And what's about to bring it back isn't going to look anything like the forest anyone imagined saving. To understand why, you have to see what this tree actually was. Before 1904, the American chestnut wasn't just common in the eastern United States. It was the forest. 1 in every 4 hardwoods from Maine to Georgia was a chestnut, trees so massive people called them the redwoods of the East, trunks 10 feet wide, canopies that fed entire mountain economies. Families timed their year around the nut harvest. Hogs fattened on it. Railroads ran on its rot-proof timber. It was less a species than an institution. The one tree the eastern forest seemed built around permanently. Then, in 1904, at the New York Zoological Park, someone unloaded a shipment of Asian nursery stock carrying a fungus nobody in North America had ever had to fight off. The blight didn't spread, it detonated. Within 40 years, it crossed 200 million acres, and the towering chestnut canopy collapsed into something almost nobody talks about. An underground network of root systems that refused to fully die, sending up doomed sprouts again and again, generation after generation, never living long enough to flower. That's the tree as we've known it for 100 years, not gone, just frozen mid-collapse. And now, the effort to finally break that cycle is forcing scientists, foresters, and regulators into a fight that has almost nothing to do with the tree itself, and everything to do with what we're willing to call wild.
Welcome to the Rewilding, where we uncover what is and what was. Subscribe if you want more stories like this.
Picture a tree so common it barely registered as remarkable, until you learn what common meant. Before 1900, roughly 1 in every 4 hardwoods between Maine and Georgia was an American chestnut, not scattered specimens. A canopy species, stretching across 200 million acres, with individual trees reaching 10 feet in trunk diameter and heights that earned them a nickname, the redwoods of the East. This wasn't just a big tree, it was infrastructure. Its nuts fell in such volume, every autumn, without fail, that entire mountain communities built their calendars around the harvest, feeding families, fattening hogs, filling railcars bound for city markets. Its wood, naturally resistant to rot, framed barns and fenced pastures, and built the railroads carrying everything else. Economists at the time treated the chestnut less like a wild plant and more like a utility, reliable, renewable, permanent. And that permanence was the trap. Nobody who depended on the chestnut in 1900 was planning for a world without it, because nothing in its 4,000-year run across these mountains suggested one was coming. The tree had outlasted ice ages. It had outlasted every native pest thrown at it. What it hadn't outlasted yet was a shipment of nursery stock that hadn't arrived at the docks in New York. The first response to the blight wasn't panic, it was confidence. In 1904, when a strange orange pustuled fungus turned up on trees at the New York Zoological Park, foresters and plant pathologists believed they understood exactly what to do. Cut a fire break, establish a quarantine zone, contain it the way you'd contain any outbreak, geographically, physically, with saws and distance. It didn't work. Crephometria parasitica doesn't need direct contact to travel. Its spores ride wind currents and hitch on birds, insects, even rain splash, moving in directions no firebreak could anticipate. Within a single decade, it had leapt the Hudson and was working its way south. Within 40 years, it had crossed 200 million acres, nearly the entire native range of the species, killing an estimated 4 billion trees along the way. Here's the part that rarely makes the summary. The containment effort itself made things worse. Desperate to slow the spread, crews felled healthy, uninfected chestnuts by the millions, hopping to starve the fungus of hosts. It didn't stop the blight. It just meant some of the last healthy giants of the eastern forest were cut down by the very people trying to save them. What survived wasn't a tree anymore. It was a root system that forgot how to stop trying. A root system that keeps trying to grow, decade after decade, is still technically alive, and that single fact is what gave scientists a foothold. If the chestnut wasn't extinct, only arrested, then in theory it could be unarrested. The first serious attempt began in the 1980s, and it ran on a kind of patience most modern efforts don't have. Breeders crossed surviving American chestnuts with blight-resistant Chinese chestnuts, then bred the hybrid offspring back to American parents, generation after generation, aiming for one precise ratio, 15/16 American, 1/16 Chinese. Enough foreign genetics to survive the fungus. Little enough to still look, grow, and behave like the tree that once defined these mountains. That took decades. Thousands of controlled pollinations, tracked across multiple tree generations, with no way to speed it up. While that program was still running, a second team skipped hybridization entirely. Researchers at SUNYSF identified a single gene in wheat that produces an enzyme called oxalate oxidase, one that neutralizes the acid the blight fungus uses to kill its host, and spliced that gene directly into the American chestnut's own genome. No crossbreeding, no generational dilution, just one borrowed gene in an otherwise entirely American tree. Both fixes looked finished on paper. Neither one survived contact with a real genome. The backcross breeding program ran into trouble first. Resistance to the blight, it turned out, wasn't controlled by one tidy gene that could be isolated and carried forward. It was linked to a tangle of other traits, and every time breeders selected hardest for resistance, they risked dragging along characteristics that had nothing to do with survival and everything to do with slower growth, weaker competitiveness, trees that could fight the fungus, but struggled to outcompete their neighbors for sunlight. After decades of work, the 15/16 American tree wasn't the clean solution anyone had promised. The genetically engineered strain ran into a different wall. Even with the wheat gene inserted, individual trees showed wildly inconsistent results, some expressing strong resistance, others barely any, a variability regulators couldn't ignore. Add to that a public deeply uneasy about releasing a transgenic organism into wild forest, and the approval process stretched on, layered with review after review. Two teams, two technologies, both discovering the same uncomfortable truth. A genome doesn't care how good your plan looks on paper. For 100 years, every version of success, breeders, engineers, foresters, meant the same target: rebuild the tree. One organism, one genome, a clone of the giant that stood here in 1900. That target was Rho-NG. What's actually taking root across restoration sites now isn't a single silver bullet tree at all. It's a mosaic. Naturally resistant wild survivors, trees that made it through the blight on their own, discovered hiding in plain sight across the old range, are being paired with back-crossed hybrids and transgenic strains, alongside a third tool almost nobody outside the field has heard of: hypovirulent viruses, essentially viruses that infect the blight fungus itself and weaken it from the inside, reducing its lethality without touching the tree at all. None of these tools on their own gets the chestnut back. Combined, wild genetics, engineered genetics, and a virus that sabotages the pathogen, they're producing something that functions like a healthy forest population, even though no two resistant trees in it are genetically identical. It isn't restoration in the old sense. It's something closer to assembly. And that reframes the entire fight ahead, because if the tree itself is now a composite, the next battle isn't biological. It's about who gets to decide whether a composite tree still belongs in a wild forest. The first open question is regulatory, and it's still unresolved. A transgenic tree, even one carrying just a single borrowed gene, has to clear federal review before it can be planted freely across public land. And that review has stretched across years, weighing ecological risk against the cost of doing nothing. Field plantings are happening now, in limited numbers on specific sites. A full, unrestricted release into wilderness is a different, much larger decision, and it hasn't been made. The second problem is one the Blighterra foresters never had to consider: climate. A separate pathogen, Phytophthora root rot, is expanding its range northward as temperatures shift, threatening to kill resistant seedlings in the soil before they're ever old enough to face the blight the fungus was actually engineered to fight. Decades of breeding and gene splicing, aimed entirely at one threat, now colliding with a second one nobody designed for. And underneath both of those sits a harder question, one with no lab result to settle it. Does a tree carrying a wheat gene still count as wild? Regulators, ecologists, and the public don't agree, and that disagreement is shaping the actual pace of the chestnut's return more than any science left to solve. None of that gets resolved by this film, and it shouldn't pretend to. But step back from the regulatory fights and the root rot and the arguments over what counts as wild, and a different pattern shows up, one that applies to a lot more than one species of tree. For most of the last century, saving something extinct in function meant restoring it exactly as it was: a pure lineage, an untouched original. Chestnut restoration is one of the clearest cases yet where that idea has quietly failed and been replaced by something more honest: functional resilience instead of historical accuracy. Not the same tree that stood here in 1900, but a population wild, bred, and engineered together that can actually do what that tree did. Anchor a forest, feed a food web, hold ground against a fungus that was never supposed to be here in the first place. That's not a downgrade. It's an admission. In a warming, interconnected world where new pathogens keep finding new hosts and climates keep shifting range maps, the untouched original may simply not be an option anymore for the chestnut or for whatever comes after it. So picture that root system again, still sending up sprouts under the leaf litter, still refusing to fully die. It was never waiting to be restored. It was waiting for us to stop insisting on the wrong kind of rescue. Once you see this story clearly, the outcomes stack up fast. A tree declared functionally extinct that never actually died. A rescue effort that abandoned the idea of a single perfect fix. A species coming back not as one organism, but as a population: wild survivors, hybrids, transgenics, even a virus enlisted as an ally, none of them identical, all of them doing the job. And a legal and philosophical fight over whether any of that still counts as wild. A fight that may end up shaping the forest more than the biology does. Taken together, they point to something larger than one species. We tend to imagine conservation as rewinding a clock, putting things back exactly as they were. The chestnut suggests that clock may not run backward anymore. What we can do is build something that survives forward. The honest question left standing isn't whether the chestnut will return. Limited plantings are already happening. The real question is whether we're prepared to call a forest wild when part of its resilience came from a lab, and whether that discomfort says more about the forest or about us. If stories like this matter to you, subscribe and join us as we uncover how species continue to reshape and be reshaped by the world around them. Somewhere right now, under 100,000 acres of leaf litter, a root system is still sending up a sprout it has sent up 1,000 times before. For the first time in over a century, there's a real chance it won't have to try again next year.